For instance, let me take the other ones. Yeah, so that is the online training. So I posted there as well. So this link you click on whenever you are on your laptop. So when you do that, uh, for instance, in on my laptop, he asks me where to download that file. I'm wondering where that link is gone. So yeah, so I just copy and paste it here. All right, so what it does was I do just enter. So yeah, so he's giving me this option. So basically it's a link that automatically download these short read data, this short read data that contains the short reads from some chromosome of some individuals. You just assemble that, and then let me take this call. So here we are. So when once I clicked, he gave me the option to save that, and you can save wherever you want to. And these are your Linux commands. Before going into that, let's talk about the today's topic. So as we said before that our subject or domains we cover are up till this RNA domain. We cover the DNA, that is the genomics part, or be it genome or full genome or partial genome or targeted genome or be it exome. So in both the cases, we are at here this DNA level, and then DNA makes RNA. The process is called as transcription. So either we stay at genomic domain or this transcriptional domain. We don't cover this translational domain because those are the proteomics people, protein bioinformaticians. So majorly bioinformatics has two big major sections. One is genomics. Obviously it covers transcriptomics also. And the people working in proteomics, they are mostly wherever you go, you can, very rare that you can get somebody having a combination of both things, because obviously both domains are big enough and they have their own challenges, right? But you being students try to get the full picture. We also covered that thing as a student, the protein part, but later on my research during doctorate was at this transcriptomic level, RNA sequencing. And later on, while when I established a group, and when we started getting the data or getting the projects for us, so sometimes it drives where the funding is coming from. So we have the projects which are on the human area or plants area, both genomics and transcriptomics, as well as metagenomics also, because it's been like 10 years. So yeah, so these are the domains which we are dealing with. So today we're going to cover this RNA thing. So before this RNA sequencing came out, there was a technique, and that technique is still valid. People are using that. So that is the microarray. Okay, microarray. This is different than microRNA. MicroRNA is basically a type of RNA. Okay, we covered that. Microarray is a technique. So what you used to do in the past, and obviously it is practical today as well, because if you have a small set of genes and you want to study their gene expression pattern, by the way, that gene expression should be at proteomic level also. right? But then people started, after this micro array came out, obviously the proteins, their structures, getting them through physics or chemistry techniques like crystallography. Obviously, it was painstaking, and obviously it was, more expensive. So we never heard of any protein sequencing technology or protein sequencers came out like the genomic sequencers are. Similarly, we are using that for transcriptomic data as well. But when this technique came out, people realized, like, rather than going at the proteomic level, because one gene can make multiple proteins, and then proteins, um, while passing through the cellular environment, are, from different organs to different places, their target sites, they get so many changes in them or modifications in them. So one gene might be, making like so many proteins. So obviously capturing each and every protein was not that easy task. So rather than that, people started studying these gene expression studies at RNA levels. Okay, and before this micro array, or probably in parallel to this, there was a PCR technique that was working. So that was qPCR, or QRT-PCR, or RT-PCR, reverse transcriptase PCR, not real time. There's also a term used for real time, for the real time data, right? As soon as the genes are expressing, their expression is quantified. But those PCR techniques, for them, the restriction was that you're gonna able to do the PCR of maybe 1 to 2 genes. But when microarray came out, and this technique was smart, so over there earlier initially they designed the slides like glass slides, something like flow cells in your NGS equipments. Those slides have different spots on them. So each spot was carrying the probes. So what a probe was, for example, you get the RNA. Obviously, probe is also an RNA, and the target RNA is also an RNA molecule. So you take them and you convert them into cDNAs, and then you put the probes. Suppose the probes are in this green color, so you just place them here. So what is the probe, by the way? This technique was actually the molecular hybridization, right? So you take some piece from the same gene, obviously that piece, the one that expresses, that makes the RNA. So you take that piece, and that piece is obviously, of some nucleotides or some bases, maybe 10 to 20 nucleotides. What is the probability of finding the same sequence of nucleotide into another molecule? So this thing goes with, there are 4 nucleotides, so we get 4 as a base, and then raised to the power the word size. So here the length which you are defining is a word, 10 or 20 or 30 or 40. So it goes like 4 raised to power 10. So there are 4 raised to power 10 different combinations, and amongst them, this combination with which this probe is made is just one. So it is 1 out of 4 raised to power 10 or 20. The probability of finding the exactly same sequence in another RNA molecule is very rare, or almost zero or negligible, right? So one, you can estimate what is 1 divided by 4 raised to power 20, right? So that's quite low. It's almost negligible or none. So these probes, they uniquely identify only the RNA pieces that are complementary to them. So you hybridize them. So you take, for example, one side of the strand, or if RNA is single stranded, so you make it second copy, right? And that is obviously the cDNA. So that cDNA can identify, that's the complementary part. And then for this RNA, you also convert this into the cDNA, and again it's a single stranded thing. So both are complementary to each other. So what you do is, you fluorescently labeled probe, in one color, suppose green, and your target RNA is labeled into another color like red or something. If you are finding a yellow spot, so that means the proportion of green and red is somewhat similar. How many probes you put? Obviously, you put like hundreds of them, hundreds or more copies of them. And if that spot stays green, so that means this red part was not that much added into it. A sharp red means where the saturation of this target RNA is more. So wherever you get the RNA, that color changes and you get the shades between green to red, yellow in between. That is the 50-50 proportion. So in this way, this hybridization technique gives us that colored pattern on that slide like thing, or later on they designed chips, right? And then we used to process them. Here we are just showing you the cross section of a chip and the probes which are there on those chips, and then target RNA is coming and there is some fluorescently labeled color attached to it, or whenever it attaches. Obviously they are all labeled red, so when they combine with the green, so obviously more red means more higher expression, so the color becomes sharp red or light red or somewhere in between. So then this thing is then translated into the numbers or numeric data, and those numbers are then processed. So that was the technique that existed before the use of NGS technologies for this RNA expression studies. These thing got famous as RNA-Seq, right? I told you like in
Excuse me, sir.
NGS. Yes, please.
Sir, we are using the single-stranded messenger RNA and then it is combining with the probe to form a cDNA, or we are using the
No, no,
cDNA.
no. Here we convert them to the cDNA because RNA is not that stable, no? So these are fluorescently labeled cDNAs. So obviously it's DNA to DNA hybridization, because RNA, you
Okay,
know
sir.
that it's degradable, it cannot survive. And
Yes.
similar happens here in NGS. Acha, what problems were happening in this microarray thing was like you are doing it on glass slides, and sometimes there is a dust on the slide, or sometimes there is a cross hybridization that is also possible, and sometimes you get some noisy regions, and then obviously you are restricted to get only the genes which are highly expressed or which are significantly expressed. The ones which are low expressed, probably we were not able to capture them, right? So what Ali Murtazawi, a postdoc in Barbara Wold's group, what he did was he worked on use of this NGS technologies for this cDNA sequencing. So he said, like, if we sequence this cDNA, obviously that cDNA is coming from RNA, right? And then you sequence that, and then you align it or map it to the reference, right? And reference can be transcriptome, or reference can be the genome. So wherever those reads are mapping or aligning, if you count those reads, obviously those reads are the counts of gene expression for that particular gene on which they are aligning or mapping, right? So RNA is captured and sequenced on NGS machines, and sequence reads are individually mapped to the source genome or transcriptome and counted to obtain the number and density of reads corresponding to RNA. And this RNA can be from known exons. So for example, if I'm only interested in differential gene expression of the genes which I already know them, so that is where those genes are recorded. So which gene, how many genes are recorded for human genome, for example, which are expressed and whose transcriptomic data is there, or transcripts are recorded into the database. So there are roughly around 25,000 genes whose data is already there. So they are already known exons, transcripts, right? So you map them, and here your objective is just to get a comparative gene expression between your sample, like patient versus normal control, or so, the sample treated with one treatment or one drug or one hormone versus another one, right? Or you are getting the samples on different time intervals, right? At different cancer stages, what is the expression of the genes, same gene across different cancer stages, or something like that. You can also check the alternative splice events. Like normally the genes, they have multiple alternative splice variants, right? So which variant is expressed under which condition? So that you can also do that. We came to know through this work of somebody that there is a major transcript, then 80% of the time that transcript expresses. The other copies are somewhat minor. So combined, like for example, we searched into soybean and one of the gene has 27 different alternative transcripts. So normally you have less genes in the database, but number of transcripts can be like 150,000 or even 70 or 80,000. So depending upon, like I am getting the transcript which is different than yours, so you submit and I submit, and in the end, number of transcript becomes higher than the original number of genes. What if you map these reads or these fragments, these sequenced RNA pieces, to the genome rather than the transcriptome, and you see that some of the reads are mapping to the region that has not been characterized or classified, right, into a known gene. So that is something which is a new candidate gene for you, right? So that's also possible. So that technique is really, really good or sensitive, and you can capture even the low expressed genes. So here is what you do as far as this protocol is concerned. So you capture the messenger RNA here. Remember, RNA-seq is not only restricted to other RNAs, but it is restricted to messenger RNAs also. Picture? So it is not restricted to messenger RNA, but you can capture the messenger RNA using this length of A's or poly A tail. That is different than ribosomal, and that is different than the transfer RNA. By the way, which RNA species is more enriched or more present amongst all those RNA within some individuals, right, or within some species? So which type of RNA is more abundant? Anybody? Any guess?
Messenger RNA.
Huh. Anybody else?
Ribosomal RNA.
Yeah, ribosome.
Transfer RNA.
Ribosome is the one
Ribosomal RNA.
that is very highly enriched. Can you tell me the reason why? Because different messenger RNAs are then formed and then they are degraded, but this ribosomal stays there because it has to do protein synthesis. And on one messenger RNA there are like many, depending upon the length of those that region, that coding sequence, or those exons, there are like hundreds of ribosomes processing the same RNA at the same time. So obviously this ribosomal is too much, and obviously transfer is also there, but I guess that should be the second highest. So he's transporting one amino acid, going back, picking up another one. So yeah. All right, so this messenger RNA is then sorted or selected and separated from rest of them with the help of that poly A tail. But again, some paper came out and they said that this poly A method is sometimes not working, because if you have that tail which is broken, what are you going to do then? So this poly A is then converted into oligo DTs, and then this second copy, or this DNA copy, is formed, because here you don't see U's but you see T's. Here you see A's, so that is actually part of RNA, and this is for sure DNA, not RNA. If it would have been RNA, there should have been runs of U's, uracils. U's and uracils are placed because they are sensitive to, or they are prone to enzymes. So enzymes are really sensitive to uracils. Whenever they spot uracils floating around in a sequence, they break. They consider that thing to be an RNA molecule, and if it is, if it has already done its work, then they simply degrade that, because we do not want to burst the the cell with overexpression of making so many proteins from the same RNA molecule, right? All right, so as many RNA copies, as many proteins are made, and this is done mostly. This is done once per that messenger RNA copy produced. So obviously there is a trade-off. So if that RNA stays within the cell, that is dangerous, So that has to be cleaved or removed. So that's why uracils are placed. So what you do next is, now this thing is cDNA, and then again you do the fragmentation and you do sequencing and then you do analysis. So this is what we are doing: isolation of messenger RNA, conversion of messenger RNA to cDNA, and then sequence the cDNA using some high throughput sequencing machine. And then obviously, when once those sequencing reads are there, you do QC, and then you read them. You take those reads and you map them to reference genome or transcriptome. And more times a given sequence is detected, more abundantly transcribed it is, right? So if we have enough sequences that are generated, that's what Murtazavi said, more than 40 million, a comprehensive and quantitative view of the entire transcriptome of an organism or tissue can be obtained. That paper came out in 2008, and obviously this paper was quite fascinating to me because when I landed at Illinois, initially I was working with a group that was working on evolution, and my project was studying the evolution on small using small nucleolar RNAs. I was getting bored of it. So till I took a course, and Murtazvi's paper that was presented to us or just given to us to read by Professor Leela Watkin. Her group was working on RNA-Seq, so probably there was the first experiment, and my second or the professor with whom I switched or shifted. She was working on RNA-Seq. She had RNA-Seq data of soybean, and then obviously I worked with that. So right away after, I guess less than 2 years, I was right on that project, right? So in 2010, I started my work on that. In 2010, I actually shifted. She had the plan before, and she had already got the funding. So I guess in 2009, probably she applied for the funding, right? But by summers of 2010, I was right there, and we this is how we started. And again, just to tell you that my story is that I took a workshop like this. So that was 3 day workshop, not on Linux or not on Unix or Ubuntu or anything. It was just about the R, so data analysis in R. So that was on RNA-Seq and microarray data. So that is where I started. So I guess next week we probably gonna do R also. But before that, we need to generate that file for R. And before that, I think we also go in Microsoft Excel, just to give you an idea some how to do that basic statistics for your data files, right? So mapping and aligning of the reads to reference transcriptome. What are you doing? And visualization on genome browsers, and we can also do this de novo assembly and quantification where we count those reads, and normalization. Obviously that normalization part is where we we normalize for unequal gene lengths as well as the unequal number of read data that is produced. After that, we're going to do this DGE testing or differential gene expression. What is differential gene expression? So how a gene is expressed across different individuals or samples or across different treatment conditions. In functional analysis, when you get a set of genes differentially expressed, we are interested to find their functions. How do they work? Right? And then after that, we can also have their gene expression data. We can put them into some gene networks, right? So this is all about the analysis part. So let's go back to our analysis. All right, my apologies. I'm sorry. I could somehow. Oh, no issues. That was my fault. It's not your fault. I somehow clicked that power button mistakenly, and then this thing got off rather than switching off my mic. So, are you guys still there? Okay. So now let me get back to my other screen, and here we can practice Linux one more time, probably one last time. Okay, and then everything will be in Windows. So get back to Windows. So that's all about Linux which we've been using. So you guys are here with me, listening to what I'm saying, students? Oops.
Yes, sir.
Yes, sir.
One person, only one person. How many of them are there in the meeting? Okay, so this Linux is starting, and again we need to do that exercise again and again. And I think no, day by day you are getting more confident in Linux, or probably you get the impression, so oh, it's that simple. It was not that, you know, earlier initially it looks uglier to you, but since once you get settled to it, आपका एक दफा हाथ इसमें बैठ जाए, and then you would say that oh, this is a lovely thing. Similar happens with R, right? So initially you're gonna hate it, but then gradually you're gonna like it, or then later on you're gonna love it, because the people who are in bioinformatics majors, if they are weak in programming, R can save you from any of the, um, yeah, let me call embarrassment, because initially when I landed over there, I tried to do each and everything in Microsoft Excel, but Microsoft Excel embarrassed me at multiple places, right? So, you have to open up that big file and do that analysis while it's open, so you are slow. And secondly, while you are doing Excel, sometimes, mistakenly your finger touches a key, and some hidden character gets into your file and it gives you the errors. We will demonstrate when we will cover this Microsoft Excel. So only two commands we need to run it in Linux. I don't know why this guy is so slow or so dumb. I'm just waiting him to return, and then I can restart maybe. So here is my
Excuse me,
Ubuntu.
sir.
Yes, please.
Sir, can we play games on Linux or not?
You can, because this Ubuntu is just like Windows, right? So here we are only using its terminal, but when you install Ubuntu, it's just like this thing, okay? Just like this thing. So now the modern day Macs and Ubuntu. So and obviously you can play easily because your whole computer and its resources are available to you. Not like half of them is occupied or taken up by Windows. You see that fancy picture up there? So that has a very heavy cost associated with it. And you see that he's loading the things here on desktops, which we are not using, right? So while you go with Ubuntu has nothing here. So in this way, you can work here. But obviously you have that graphical version of Ubuntu if you install it. So people who are trying to make dual core, they will see it's exactly the similar thing. Or probably remind me, we can log into the next class through one of our big machines that are all Linux based, and things are there. So there is something similar to Microsoft Excel. There is something similar to Media Player there. Obviously not the ones which are the property of Microsoft, like MS Excel and MSPP PowerPoint. Other than that, they had LibreOffice, LIBRE Office. And for PPT, there is another thing for presentation, I guess, Libre presentation or PPT something. So that's the tool where you can do exactly the same things which you are doing on your Windows machines, right? So what we need to do when we start, anybody? Give me the command. First thing I gonna do is, so I'm
CD,
very good.
MNT,
Why?
then
CD,
Why?
MNT
then
to
What?
log into the Linux
then
system, switching between Windows and Linux.
Yeah, and वो तो मैं switch कर लिया. I switched, but now I want to
then
access is actually belongs to Windows, right? So he's not given the access. So he need to
sir, it
probably, yeah, he need to probably
the co-
set his root or way. to approach those files. Okay. So you have to work in someone else's house or in the kitchen. So you go there and first you wanna, ask him where is the kitchen, or you go straight to the kitchen, or if you know about it, so you need to just reach there. Okay. So in order to get access to those files which are present on C drive, and C drive is not available to Windows, so what you're gonna do is CD, sorry, is not available to Ubuntu, and then you do MNT. So we know that this thing is within this folder called as MNT. I just put this dash thing danda, and I press enter, and he will give me different options. If I don't put danda dots and he has dot, these are the other things, something else, right? So I just need to put that slash thing, and then I put M because M for media and M for MNT, so I need to put another letter here. This thing is now for sure going to which folder? ME will go to, if I press tab here, he will lead me to media. But what if I write MN? So obviously he has no other options, but if two letters are here used and one is MN, one is M, one is N, so he will straight away go here. If you're using M plus E, that is ME, then obviously he will go here, right? So, and then within that MNT again, you press tab and he says there is an C here, and then there is, within C you have kya tha ji? You press just tab and he will give you
Users.
Yeah, users.
And then Wasim header, and then desktop, and then NGS.
कौन बोल रहा है? Right, I'm
Eman
not.
Abdullah.
ये अमन, thank you so much. जीती रही. क्या कर रही हैं आप आजकल?
Sir, PhD biotechnology
From?
from NIPG. But मेरी research जो है वो Germany और Denmark से है.
Very nice. So you're working on probiotics, Arslan Bhai's student?
No, sir. I'm working on the genetics, neurological disorders.
Yes, yes. It is a wonderful organization. So Wasim Heather, and again within tab, where we need to go desktop, right? And you also set your path, and within desktop, the folder was online trainings or NGS. So folder was NGS, and that's it. So we reach to the kitchen. Now we need to make that biryani out there. So we can do Control and L together, and duster will wipe everything. Up arrow key will repeat the previous command. Pressing it twice will be the further previous, and one more time another and another. So whichever commands you actually ran, they are stored here in its memory, so you can reuse them, right? If you click up arrow key, you can go to previous, and if you press the down arrow key, obviously you can go towards the present. So newest commands, up arrow key for the older commands, and so on, right? All right, so I want to see what I have in this folder, ls. Okay, and I want to need two files. Number one is this mu1.txt, that is my sample, and transcripts. Where is transcripts? There is a file called as at, that is Arabidopsis thaliana underscore transcripts.txt, right? And this data is coming from Arabidopsis, but at the same time I gave you one human data also that you can also practice. So for that we will use that data set for variant calling. You can also do genome assembly. Okay, the people who are asking for practice, just take that data set which we highlighted over there, download that and put it on your laptop, and then what you do next is do QC and do assembly and see what sort of context you are getting, and then tell me that from which source this read data is coming from. That's our assignment. Okay, also discuss their quality QC reports. All right, so that file is at underscore transcript. So let me see how this file looks like. So I can go with this head and at underscore transcripts. When you press tab, so that comes. So these are just sequences in FASTA format, the reference transcripts, and they are longer than that. Okay, whereas the sample is, I can just see that head over mu1.txt. So that's these short read files. So all what we need to do is you take that read and map it wherever it's mapping or aligning. And obviously these are all 100 nucleotides, they must be matching. You are allowed to do only 2 or 3 mismatches, not more than that. Here we are not doing the local alignment or global alignment which we covered in our bioinformatics class. So those kids, this is not the local or global alignment. This is the thing which we did in pattern matching. So pattern matching may you must have exactly the same thing, right? For example, if a long sequence you want to get a promoter, and if it is a prokaryote, so obviously we know that it should have a Tata box. So that Tata box is finding Tata box in a sequence is something which we call it as a pattern finding, right? Start codon. If you're just looking for start codon, again it's a pattern matching job, matching job, and you want to have the exact matches, exactly the same thing or identical things, or what you can do is allow 1 or 2 mismatches, so approximate matching, so approximately similar, so exact match versus approximate match, but not the local alignment like BLAST. Why don't we use BLAST here? Because what BLAST does is it will take the original sequence and break it into those small fragments, right? Or chunks, and take the reads and again break them into small chunks, and wherever you are getting the highest pair in terms of any piece getting from read and any piece getting from the reference, that pair will be reported. That we don't want actually. All right, so there is a tool called as bowtie which we use here. So one of them is where is that? Did I install bowtie? I think I did not, so I need to go sudo apt क्या था जी? Install bowtie2. Bowtie, yeah, bowtie2. Right? I think it's there. Is not it? Anyways, so let him come. It's a small tool and it will come real quick. He says it's already there. So you just do that and you get that bowtie2, and there is simple bowtie as well as bowtie2. The latest one is bowtie2, and I think we were using bowtie2. The commands, let me open them and place them here on the desktop. And yes, and these are your commands. So I'm waiting. Where is that document? So he's opening each and every document which I used to open because it accidentally closed. So it has a bad habit of opening everything. See, that's another downside or bad thing about Windows. I did not ask him to open everything which I was opening because that is the uncall for help. Don't do it. Okay. If people work on your advice, you give them a free advice. If they if they pass, or if they are successful in getting that target through your advice, they will not gonna appreciate you, right? And if they are failing that, obviously they will definitely gonna blame you. The source is not myself, but who said this thing? Anybody? You might have seen that on social media nowadays, and we realize that he's quite a sensible person, right? So one gentleman, he's saying is his dialogues are nowadays, so one of those quotes. Anyone guess who said this thing? He just won the American elections. So what we do is copy and Mr. Trump. So what you do is, so that is a very wise saying. Don't give free advice to the people, and he gave the other tips how to become a successful businessman or money maker or a successful person in your life. So those start clips are around on the media. Bowtie2-x 80 indexes. Oh, I'm sorry, that's not the command I wanted. Just want to run the indexes first. So two things I need to do. I need to use the tool called as bowtie build, right? Bowtie2, and there is dash. This tool is build, 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 and that means build indexes from which file. Let me tell you what these indexes are in a moment. 18 dot underscore transcripts dot txt, and name them as, and I give them a name like 80 underscore indexes, right? So he will make the indexes from those scripts. So have you ever used libraries? So when you go there, in our times, we had in the library the sections where you had the indexes. So there were small cards placed from the books with A go to that cupboard, or the books from, starting with double A go here and AB go here, and then there, if their second letter is AC, like accounting or something, they go here, and something like that. They are placed properly in a library. So here the problem is that you can take a short read and you can match into multiple places, and you can do that against all sequences, but that's gonna take really long. So what happen is you make their indexes. They are there in machine language. Here we get some few files. Here it is 80 underscore indexes dot one dot two three four, and then dot reverse dot one dot bt2. bt2 means they are coming from bowtie2, and indexes is the name which we gave to one, and one or two for reverse indexes, and four files for forward indexes. So what's happening over here is your data is divided into four different groups or sections. So he takes the original sequences, those fastest sequences, and then tries to find out the common sequences amongst them. So those are the ones which look like similar. Those, the original reference is then broken into short fragments or sequences, right? And then he takes a read, and obviously on one side of in file number one, for example, I am taking the sequences which starts with ATG. So now your read fragment starts with ATG. So I don't need to search them for rest of the five files. I simply go and put it into the chunk from where that common pattern is coming from. So that will make my search space easier, right? If it is hard to understand, just remember, like, this is just a technique used by bowtie2 to make it search quicker and efficient. Okay. Right, so now you have made those indexes. Next step is you just call the bowtie2 tool, and that is just for alignment. And then you need to give him the index file with just our option dash x. Dash x means these are my indexes. What is the name of the index files I wrote? I wrote it 80 underscore indexes. You just remove that dot thing. इतना नाम देना है अपने, and then you give him the name of the data file or the read file which you want to align onto those indexes, right? So mu1.txt, and you just enter, and something happens over here. So again, those read fragments are here, but rather than having those quality scores, we were having some other things like static, like 4 and static and 0 and 0. That's the output file which we are seeing. So when it aligns, it gives us those reads, but in the same time it adds some additional things here in the output. 16 means that this read has aligned somewhere. 4 means it did not align. Static means no gene on which this read was aligned, or no transcript on which this read was aligned. 000 static because this thing was not aligned with anybody. But this thing, that is this flag, this thing is called as flag, so that is flagged 16. So flag 16 tells us that this thing aligned somewhere, and this is the transcript on which this read has aligned, and it has aligned from 371 position onward, right? So that reference gene or transcript, its nucleotide number 371 से लेके up till nucleotide number 470. So 470 tak it would have been aligned, and there is another thing that is 42, and this is just 100M. So this just good quality. This is another scale. So it's telling us like this alignment is pretty good quality. You can also see if there is some mismatching, or if there is some something that is there here. So if some nucleotide is mismatching, only one or two mismatches are possible, so they are also reported. They are important to capture our SNPs, right? So those mismatches are also important. A couple of one or two or more, not more than 3 or 4 mismatches are allowed here, but those mismatches are also very important for us. Okay. Right, so this is how we aligned this read file, and since we aligned and we just displayed on the console, but nothing you can do about it unless you store it. So how you can store that output file? So you again write this command, but you direct your output into a file. So that greater than sign means anything that is the output of this command on the left side, place it into this file. I just write that file name as mu1 dot or underscore output. So that's my output file. Okay, so that's the file. So now he's not writing anything. Acha, he gives us a statistics. He said that 1000 reads were unpaired, and they were used, and they were single reads, not paired ones. 876 aligned 0 time, they did not align, so alignment is quite low. Normally in the real data this is higher, so 87% is here and 6% or whatever that. 68 amongst them, 6.80, they aligned exactly at one time, but there are the ones which are aligning at multiple positions, so they are more than one time. So 56 reads are aligning at multiple positions, right? So overall the alignment percentage is 12.4%, adding these two numbers. So 12.4% is overall low alignment, but as I said, this is just a test data, just to test that. But in real life these figures are reversed. So what I have seen in my data set is these alignments, depending upon how good your transcripts are reported into the databases. So initially when I started this soybean analysis, this first version of this phytosome was a database where we had that plant genomes and transcriptomes. So on the first version of soybean, this the first assembly, that was having lower alignment percentages because the number of transcripts was less. It was around 40,000, but in the second variant they increased to 70,000. So obviously I got more search space or more space to search, so obviously those, they got more in numbers, right? So this is how we align those files. ls can tell us where our output file is, and now you can go here and take that output file. So I think I gonna stop right here, but just to show you this file, and obviously we're gonna repeat that thing in our next class. So I can go and open with, choose another file. How can I go? I think I can direct B, or I can just open it here. Somebody is making noise. So that format is called as SAM, sequence alignment and mapping, right? But in machine language, later on we convert it into BAM. So some of the tools they directly convert it into BAM files. So they are just ugly characters, sterics and squares and cubes and anything like that. But here you can see the ATGCs, so that's SAM format. So here he's giving us those gene IDs, and then he's talking about their lengths. आपकी क्लास है?
अगर कैट वाले बच्चे उसी में बैठेंगे आपकी क्लास
क्यों? अच्छा
ये तो बिठा दो, मैंने को बिठा दो।
चलें, आप ये यहाँ से ज़रा ये क्लोज अप करवाएं, और ये मैंने आउटपुट दिखा दिया इनको. एंड देन व्हाट यू कैन डू फ्रॉम हियर इज यू कैन सर्च वेयर इज दैट 16 थिंग. सो दैट 16 मीन्स we are actually seeing the ones which are mapped, not this one, but I just want here. Yeah, this one is matched here. So one read matched on this scene. I can just simply count how many reads are aligning to this scene, and next part is there is a code, Python code, so that can gives us these counts. So there is just one occurrence, and second, is it the same, and third and fourth and sixth. Yeah, we just keep track of that read ID. I think 7 or 8 reads are aligning to this gentleman. So again we start from here. Number 1, nishani iski ye rakhne ki it is 6 7 7 onwards. So then 2 is here, and then 3 and 4 and 5 and 6 and 7 and 8 and 9, and so 9 occurrences are here. So that means 9 reads are mapping to or belonging to this gene, and later on we can just use that Python script, right, which is there to count how many reads are aligning to this part, but I will do that next time. So up till now, if you have any questions, let me know, but I will repeat this part. But before we go into the next week, The sample I gave you, just do this QC and assembly thing with that and try to find out that which sample was given to you, right? It's coming from which individual and which chromosome and which part of the genome and what genes it carries. That's your homework. Okay. All right. So see you next week. Any questions, concerns, queries, let me know. Sadia is also here. You can take that. And let me stop sharing and let me stop.
Recording?
Yeah. How can I stop? You started. I think you
No.
can. Missed. Yeah, somehow I got out of it.